Hi everyone, today we're going to have a look at the process of photosynthesis, which kind of divides itself into two stages. First, we're going to have the light-dependent process, and second, we're going to have the light-independent process. In this video, we'll mainly focus on the light-dependent stage of photosynthesis, so let's get right into it. Now, photosynthesis, as I've explained before, divides itself into two stages. First, we have the light-dependent process. And second, we're going to have the light-independent process. Now, I kind of like to say that the light-dependent process divides itself into mainly two sorts of stages. First, we're going to have photolysis. As you can probably infer from the word photolysis, photo meaning light and lysis meaning breaking. In this context, the breaking of bonds. Photolysis essentially means using light energy in order to break water, but this I'll explain further later on. The second part of the light-dependent process consists of using the ETC, as we mentioned earlier from our earlier videos, and this process involves the creation of ATP and NADPH, and also hydrogen ions. Now, the light-independent process kind of sums itself up into the cycle called the Calvin cycle. This we will be speaking about in another video. But first, let's look into the light-dependent process of photosynthesis. So the light-dependent process, where does it occur? Well, it occurs in the thylakoid membrane. And the thylakoid membrane is kind of in the chloroplast. Which is, in a larger context, it's located in the plant cell. And what happens is the thylakoid membrane kind of provides this sort of a division barrier between the stroma and the thylakoid lumen. And what happens is we're going to have these little proteins called photosystems. Now, in these photosystems, we're going to have a lot of pigments that are embedded in the photosystem. If I were to visualize photosystem 2, which is kind of where our entire process of the light-dependent process kind of really starts, photosystem 2 would kind of look like this. And we're going to have pigments embedded in the photosystem. Now, at the center of the photosystem, there's going to be a special pair. Now, the special pair, this is literally just two chlorophyll molecules. So as you can kind of visualize in these photosystems, in photosystem 2, we're going to have pigments, and then we're going to have a special pair, which is just two chlorophyll molecules. Now, each of these chlorophylls can literally have one electron in the center. So it's kind of going to look like pretty much like this, and this is going to be the electrons, which are located in the center. Now, when these pigments kind of absorb the light energy, this light energy gets converted into what we call chemical potential energy. This is going to become chemical potential energy. And now having chemical potential energy kind of really increases the overall energy value of these individual pigments, and it's going to make these pigments very, very unstable. So because these pigments absorb light energy, which is later converted into chem potential energy, this is going to increase the overall amount of energy, and that is going to make these pigments very unstable. Now, please do not confuse the state of being unstable with having with being excited. We normally use the terminology excited for only electrons, and if something has a lot of energy, especially in this context of pigments, we're going to call them unstable. So we're going to have unstable pigments. Now, because these pigments become unstable, these pigments are going to pass on the chemical potential energy to their neighboring pigments. So what happens is, due to overload of energy, these pigments become unstable, and in result, they pass on their chemical potential energy to other pigments. Now, this process kind of occurs and repeats until chemical potential energy all of the chemical potential energy kind of entirely accumulates in this special pair. Which is again at the center of the photosystem. So as we can see, we start with the top pigment, which gets a lot of light energy, absorbs light energy, and then it turns it into chemical potential energy. And because it has so much energy, it's going to become unstable. Therefore, it passes on the chemical potential energy to the next pigment, which passes it on to the neighboring pigment, and then so on and so on and so forth, until all of that chemical potential energy kind of accumulates in the special pair. Now, this accumulated potential energy, chemical potential energy, are going to be used by electrons in the special pair. Remember how I mentioned that there are electrons in the center of the special pair for each of the chlorophylls? Well, these electrons are going to use that chemical potential energy. And these electrons, remember what I said about electrons having a lot of energy? Well, they're going to be excited. This process, wherein these electrons, due to high chemical potential energy, become excited, is called photoactivation. Now, after this process, we have electrons, which are in each of these special pair molecules, which are chlorophylls, that are excited. Now, when these electrons are excited, we essentially have these electron carriers that are found in the thylakoid membrane, and they accept these excited electrons. We're going to have electron carriers that are embedded in the membrane, and they accept these excited electrons. And these electrons, through the electron carriers, are kind of going to take this sort of journey, take this sort of path where they move into photosystem 1 from photosystem 2, where it was excited, and then later kind of ejected and escaped. Now, by the time that the electron kind of moves towards photosystem 1, the energy of the electrons are kind of used by the hydrogen ion pumps. And this is very important. The hydrogen ion pumps kind of pump the hydrogen ions from the stroma to the thylakoid lumen. And now this is going to kind of create a proton concentration gradient. So kind of just to provide you an overview of what happens, we have electrons that are excited in photosystem 2. Via electron carriers, they take this sort of path towards photosystem 1. In this path, they encounter hydrogen ion pumps, which use the energy of the excited electrons in order to pump the hydrogen ions from the stroma to the thylakoid lumen. And because it pumps the electrons, pumps the hydrogen ions via active transport, this entire process is going to increase the concentration gradient, kind of create this proton concentration gradient, and therefore we're going to have a lot of hydrogen ions in the thylakoid lumen. And beware, the thylakoid lumen is a very, very small space. So the concentration gradient is going to become really steep really fast. And now that's where the ATP synthase kind of comes in. Now, as you can probably see by the ending A's, this is an enzyme. However, in this context, we're not exactly going to look at its function as an enzyme. We're kind of just going to take it as a sort of as a sort of a protein that can transport these hydrogen ions according to their concentration gradient down their concentration gradient. And this process is really important. This process of ATP synthesis by ATP synthase, wherein the hydrogen ions that were in high concentration in the thylakoid lumen move down the concentration gradient and back into the stroma, this process releases a lot of energy. And this energy is stored in the form of ATP. And how does it do that? Well, this is because the ADP molecule, essentially we add a phosphate ion to it, an inorganic phosphate, and then we're going to get the ATP molecule, which is going to store the energy for us. So that is exactly why we say that this process of the light-dependent process of photosynthesis creates ATP. All right, that is pretty much it for today. In our next video, we're going to take a look at the process of photolysis, which is very important because it's kind of known to supply these hydrogen ions that are involved in this entire process. And we're going to also take a look at this process of creating NADPH and also other hydrogen ions.